Hello all, welcome to my channel Pharma Dive. This channel is purely for educational purpose and aims to provide valuable information. So, if you are ready to dive into the world of pharma, hit that subscribe button and join the journey. Let's explore, learn and gain together. Today's topic is autoclave in pharmaceutical industry. Sterilization by moist heat under pressure. What is autoclave? An autoclave is a sterilization equipment that uses saturated steam under high temperature and pressure for a specified exposure time to destroy bacteria, viruses, fungi, and spores. It is widely used in pharma industry to sterilize equipment parts, glassware, culture media, garments, rubber closures, certain drug products, and many other components used in sterile manufacturing. What is the principle behind it? Autoclave works on the principle of moist heat sterilization, where saturated steam coagulates the microbial protein, leading to the destruction of microorganisms and their spores. Let's understand the principle of autoclave. An autoclave works on the principle of moist heat sterilization, where saturated steam under pressure raises the boiling point of water, allowing high temperature steam to penetrate materials effectively. When steam condenses on cooler surfaces, it releases latent heat that denatures the microbial proteins and enzymes, resulting in the destruction of microorganisms including bacterial spores. Standard sterilization conditions are 121 degrees Celsius at 15 psi for 15 to 30 minutes for standard sterilization cycle and 134 degrees Celsius at 30 psi for 3 to 5 minutes for flash sterilization for medical devices. Let's see components of autoclave. An autoclave consists of several essential components including the sterilization chamber, steam jacket, door, steam condenser, heat exchanger, vacuum pump, pressure gauge, temperature sensor, safety valve, alarm system, control panel, and drain system. These components work together to ensure safe, efficient, and reliable steam sterilization. Now, let's explore the different types of autoclaves used in pharma industry. Autoclaves are classified into vacuum, pre-vacuum, superheated water, laboratory, and double door types. The selection of an autoclave depends on the process requirements, product characteristics, and load type. Let's see step-by-step working cycle of an autoclave. The autoclave sterilization cycle begins with loading the material, followed by air removal to ensure effective steam penetration. Saturated steam is then injected and the load is held at the required temperature, pressure and time to achieve complete sterilization. After the exposure period, the steam is exhausted. The load undergoes a drying phase and the sterilized materials are finally unloaded. Critical process parameters are the effectiveness of the sterilization cycle depends on maintaining the correct temperature, pressure and exposure time throughout the process. Autoclaves are widely used in pharma industry for the sterilization of culture media, pharmaceutical products, glassware, equipment parts, garments and accessories. They are also used to sterilize rubber closures and for the decontamination of biohazardous waste, ensuring product quality, regulatory compliance, and patient safety. The effectiveness of an autoclave depends on maintaining critical process parameters, including temperature, pressure, exposure time, and the use of high quality saturated steam. The temperature should be between 121 to 134 degrees Celsius. The pressure should be between 15 to 30 psi and the exposure time should be between 15 to 30 minutes. Continuous monitoring and control of these parameters ensure effective sterilization, consistent process performance, and regulatory compliance. Let's see validation of an autoclave. Before an autoclave can be routinely used, it needs to be qualified and validated. Validation confirms that the autoclave consistently achieves sterility. The typical validation activities of an autoclave include installation qualification, operational qualification, and performance qualification. Heat distribution and heat penetration studies during validation are critical. One of the most important aspects of autoclave operation is routine requalification. Let's understand why it is essential. Routine requalification includes a series of tests to verify that pre-qualified autoclave continues to perform effectively, consistently, and in compliance with regulatory requirements. The autoclave requalification activity may include the following tests and applicable to all pre-qualified autoclaves. The first test is steam quality test. It determines the level of non-condensable gases and moisture in the steam. The second one is vacuum leak test. It is conducted to evaluate possible air infiltration into chamber under vacuum. Third one is Bowen-Dick test. It is conducted to evaluate the effectiveness of air removal from all parts of sterilizer at working temperature and pressure. Fourth one is air detector test. The air detector should cause a fault to be indicated if the amount of air or no condensable gas in the chamber. Next one is HPHV load, that is empty heat distribution study. It is a testing process that demonstrates the sterilizer is capable of achieving a uniform temperature distribution throughout the empty chamber and identifying the cold spot. Next one is HPHV load, that is loaded heat distribution and heat penetration study. It is a testing process which demonstrates that the sterilizer is capable of achieving a uniform set temperature throughout the chamber in distribution and penetration condition and holding those temperatures during the exposure period of cycle and specified sterilization conditions. Let's see sterilization indicators. Sterilization effectiveness is monitored using physical, chemical, and biological indicators. Physical indicators monitor temperature, pressure, and exposure time, while chemical indicators such as indicator tape and integrators verify that the required sterilization conditions have been achieved. Biological indicators containing Geobacillus stereothermophilus spores provide the highest level of assurance by confirming the effectiveness of the sterilization process. Let's see advantages of autoclave. Autoclave provides a reliable and highly effective method of sterilization, including the destruction of resistant bacterial spores. They are non-toxic, economical, environmental friendly, and offer excellent steam penetration, ensuring efficient and consistent sterilization of pharmaceutical materials and equipments. Let's see limitations of autoclave. Autoclaves are not suitable for heat or moisture sensitive materials and may cause corrosion of certain metals over time. Their performance depends on high quality saturated steam, proper drying to prevent wet loads, and a well-maintained system to avoid vacuum leaks that can compromise sterilization effectiveness. Let's see regulatory requirements. Autoclaves used in pharma manufacturing must comply with GMP requirements, validated sterilization procedures, and approved SOPs. Compliance also includes maintaining cycle logs, biological indicator records, environmental monitoring data, and documented periodic calibration and preventive maintenance to ensure consistent sterilization performance and regulatory readiness. Regulatory agencies expect documented evidence of sterilization effectiveness. Autoclave failures are major compliance issues for sterile manufacturers. Before we wrap up, let's quickly review the key takeaways from today's presentation. Autoclaves are essential sterilization equipment in pharmaceutical manufacturing. The use of autoclaves in sterilization of equipment, components, and other manufacturing materials in sterile pharmaceutical production is important since they are capable of supplying an effective and dependable method for providing steam sterilization. The functioning of an autoclave is based on efficiently altering the latent heat from saturated steam to microorganisms. Thus, they provide rapid death to microorganisms. Proper operation, validation, and monitoring ensure product sterility, patient safety, and regulatory compliance. Thank you everyone for your time and attention. If you found this presentation useful, please don't forget to like, share, and subscribe to Pharma Dive for more pharmaceutical concepts.